is go over the last worksheet, photosynthesis and energy worksheet. So I'll share my screen with you. Can you see it? Photosynthesis and energy production worksheet?
Yes.
Okay. So just go over each of the questions sequentially. And indicate which ones people seem to get hung up on. First one, not so much. 3 processes by which a cell can generate ATP, photophosphorylation, substrate level, and oxidative phosphorylation. We're going to spend a lot of time today on the latter 2, substrate level and oxidative phosphorylation, and see where and how they are involved in energy production. Photosynthesis is a source of energy for most of life forms. There is an alternative, and that is chemosynthesis. And most of you got that right. Now, the next one, I had a preferred answer. There's another answer that many of you chose that is also not incorrect. Now, what I was looking for is that photosynthesis consumes energy and is anabolic, whereas respiration releases energy and is catabolic. About half of you put down endergonic and exogonic. And as I said, those are not incorrect, so I gave full credit for those. But I preferred the former terms because these processes really don't have much to do with heat, whereas the other terms imply that heat is involved. Okay, number 4, in oxidation reduction reactions, which statements are true? Electrons are transferred back and forth between carrier molecules. A substance that gains an electron is reduced. The next 2 are not correct. Substance that loses an electron is oxidized. These reactions occur in pairs. And the function, the whole point is to release energy for ATP synthesis. Now, number 5 is about this mnemonic oil rig. Is it true that it stands for oxidation is loss and reduction is gain? And it is true. I do find this handy when thinking about reactions, whether a substance is oxidized or reduced. Next one, evidence occurring during photosynthesis and respiration take place in particular subcellular compartments. Match them up. So absorption of light is number 5, the thylakoid. Calvin cycle is 6, the chloroplast stroma. Photosystem 1 is again the thylakoid. Krebs cycle, mitochondrial matrix. Glycolysis is in the cytoplasm, and oxidative phosphorylation is in the inner mitochondrial membrane. Chemosmosis is a critical step in ATP production. Indicate which items are true. Well, it generates ATP by passing ions across a semi-permeable membrane. It is not a catabolic process, it's anabolic. The source of electrons is the electron transport chain, that is true. And an ion concentration gradient is essential. And we're going to talk a lot more about all of these processes later today. Then number 8.
Professor,
Yeah.
on question 5, sorry if I missed it, but who is Scott Pittman? Was that just a random name?
He's no, it's not a random name. He is a learning assistant in some of the courses in Bio 130, not in this.
Okay. Because that kind of tripped me up. I was like, who is this Scott Pittman person? Like, okay.
Yeah. As though that was part of a trick in the question. No, it's somebody that you've never met and probably never will.
Okay.
Okay, Calvin cycle and Krebs cycle. Each of these processes belongs to one or the other. I think only 1 or 2 people got all of them correct. Most people missed 2 or 3. Anyway, it requires oxygen, and we'll talk a lot more about oxygen and lack of oxygen today. That's the Krebs cycle. Creates glucose is Calvin, occurs only in plants is Calvin. Produces ATP and carbon dioxide, that's Krebs. Anabolic, Calvin, in the mitochondrial matrix is Krebs. Part of photosynthesis, Calvin, oxidizes glucose, Krebs. Includes the chloroplast dark reaction, Calvin. Part of aerobic respiration, Krebs. Occurs in the chloroplast stroma, Calvin. Consumes ATP and carbon dioxide, Calvin. And is a catabolic process, Krebs. So most of you got most of those right. Most people got a couple wrong. All right, then I gave you a little story about seeing a tree that was yellow and you thought it was dying, but then you saw it was growing and so you knew it wasn't dying. And how can you explain this? And many of you said something about it producing chlorophyll, that's why it's growing, but it also produced a yellow pigment that masked the chlorophyll, and that's why it looks yellow. So another possibility would have been that this is a parasitic plant. So, but thinking about it, thinking of yourself out walking along and you see a tree that's yellow, why would you think it's not a parasitic plant? You're looking at a yellow tree.
Well, no, you go ahead.
Oh, like when seasons change, I think the chlorophyll is depleted, or it's something.
Chlorophyll depleted.
Like something with the season change, like something's depleted in the leaf.
Unlikely. You're thinking of maybe like a nutrient deficiency and it's causing the plant to run out of gas.
Oh, I think on the last test there was a question on why the plant would change colors. Is that what you're asking? I'm sorry.
Yeah. We expect plants to be green. Most of them are, but when we see one that isn't green, then what's the explanation? And what I'm asking now, I've said that probably this yellow tree is because it's got a yellow pigment that's masking its chlorophyll. And so I'm going further and saying, all right, why would you say this is not a parasitic plant?
Professor?
Yeah.
Didn't we mention earlier that a parasitic plant would have something attached to it that is taking the color? So wouldn't this be a yellow tree because something has attached to it, making the pigment change?
A parasitic plant is just that. It's parasitic. It's entirely dependent on its host. So if you recall, I showed you pictures of these little structures called haustoria.
So you'd expect to see more attachment to the tree, not just the tree itself. There would be additional
Yeah, and so if
plant
you're,
on there.
looking at a tree, it's got roots, right? And if it's parasitic, it wouldn't have roots. It's getting all of its sustenance from its host plant. So it's very unlikely that if you're seeing a yellow tree, that it's a parasitic plant.
Professor, I have a question.
Uh-huh.
So do you mean that the tree that is yellow have to be contact with the tree that is green, right? It doesn't have to be the same. It can be another
If it's
different type of tree.
If it's parasitic, it would have to be attached to the green plant. Yeah.
And it doesn't have to be the same plant, right? It can be different type of plant.
That's
Just
right.
need to be green.
They're distinctly different plants, but not independent of each other. They have to be in physical contact if it's a parasite.
Okay.
All right, number 10 is energy currency is ATP. Then this next question, I actually made an error in wording it. During aerobic respiration, glycolysis yields 4, and I say 4 net ATPs. It should be 4 gross ATP molecules. And the Krebs cycle yields 34 for a total of 38. So since I made a mistake on wording that, I gave you credit for however you answered it. Next one's about lions.
Could you go over that one, Professor? Like
Yeah.
the slide for that one?
Yeah, we're going to talk about this.
Okay.
We're going to have stoichiometry later when we get to the slides. And so,
Okay.
I hope to make it crystal clear what's net and what's gross. Do lions need vegetation? And the answer is yes. Not that they eat plants, but that they eat prey who eat plants. And most of you got that. Respiration and photosynthesis are often portrayed as having opposite functions, which they do by and large. They also have similarities. What might they be? Dependency on enzymes to do their jobs, occur inside a membrane-bound organelle. They both do. Most of you did not get the third one, occur in green plants. And that's an important point because you have to remember that plants respire too. Not only are they green, not only do they make food, they use food. And they use the food that they themselves make. So both of these processes occur in green plants. Feedback inhibition is a term that designates an unusual class of enzymes. No, that's not true. We've talked about feedback inhibition as a type of regulatory mechanism.
Professor,
Yeah.
on the green plants one, the question I had, because I know the difference is like breaking down of energy and the other one's kind of the like storing of. So how does both processes work in the green plants? Like I get the photosynthesis, but not so much the respiration for
Yeah.
occurring in green plants.
Okay. We'll talk about the respiration part of it later in
Okay.
the slides. But in a nutshell, green plants have mitochondria. So photosynthesis goes on in the chloroplast, and that's how plants make food. But respiration goes on in the mitochondria, and that's how plants use food. If plants only had chloroplasts and not mitochondria, they couldn't use the food that they themselves make. Yeah, so we'll talk about that a little later. Chlorophyll is a plant pigment that captures energy by absorbing red and blue wavelengths, but not green, which it reflects. Photosynthesis can occur in any part of a plant. That's not true. The plant part is not green, it's not photosynthetic. Main components of a chloroplast are, there are 5 of them: thylakoid, the stroma, the grana, the outer membrane, and the thylakoid compartment. And finally, chloroplast harvests chemical energy from light waves. It does that through the activities of not 1 light reaction, but up to a maximum of 3 light reactions. That's false. Why is it false?
This is just a guess. This is Sierra. It's because my guess is that there's no maximum, then it just can continue to do that over and over.
Yeah, there is a maximum to the number of light reactions, and it's not 3.
I was kind of on the same thought process that she had as well. I was unable to locate the information of what the max number was.
Okay, I don't have my slide set on photosynthesis up here, but if you remember, when a photon of light comes in, it first strikes photosystem 2. There are 2 photosystems. And
Okay.
photosystem 2 intercepts the first photon, excites an electron that goes, has a high energy, and then it goes through an electron transport chain, loses a lot of energy, and then another photon comes in to photosystem 1. Photosystem 1 comes after photosystem 2, and that excites the electron again, and it goes through some other carriers and finally ends up in the dark reaction in the Kelvin cycle. So the reason that's false is that the maximum number of light reactions is 2. Okay, anything else on the quiz? One other thing I should mention, I think on what you saw in your worksheet, it said it was worth 20 points. It's really worth 19. Okay, I'm going to stop sharing that. We're back again to our overview. Where we're going, and now it's mostly where we've been. We've talked a lot about energy, and enzymes, and photosynthesis. We had a question and answer period last time about photosynthesis, primarily photosynthesis and cell division. And now what's left, we've talked some about respiration, but we're going to go into it today in a lot more depth and address some of the questions that you've already asked. How do we derive energy from food? That is by way of respiration. And there are 2 types: aerobic, which occurs in air, and anaerobic, without air. The distinction is very important, and we'll go into the detail about that. The cycles, aerobic and anaerobic. Aerobic respiration involves glycolysis, hydrolysis of glucose. It involves the Krebs cycle, then electron transport, which feeds electrons into oxidative phosphorylation, which is chemiosmosis. Without oxygen, it's anaerobic, and that's when we get fermentation. And this can be, it can be one of 2 branches. Depending on where it occurs, if it occurs in microbes, for example, a yeast, glycolysis produces alcohol and carbon dioxide. If it happens in a muscle that's working hard, glycolysis produces lactic acid, and that has unpleasant consequences for we humans. So again, this is an introduction, and we'll talk more about these in detail. The overview of respiration involves primarily the mitochondrion. And again, I emphasize mitochondria are present in both plants and animals, so they both respire. Respiration starts with glucose and glycolysis. And that process, it's a very long, it's a nine-step process that leads to pyruvate. I'll show you that later. And pyruvate then can leave the cytoplasm where glycolysis occurs, cross the mitochondrial membrane, and then provide the material to start the Krebs cycle. Glycolysis produces electrons carried by an electron carrier, NADH. It also has a substrate level phosphorylation, which produces ATP. These electrons that are produced by glycolysis go into the electron transport chain. But the pyruvate goes into the Krebs cycle and produces a lot more electrons that go into the electron transport chain. Also in the Krebs cycle, there is another substrate level phosphorylation, produces another ATP. These are relatively minor amounts of ATP. The major source is oxidative phosphorylation, followed by chemiosmosis. That produces the bulk of ATP. And Patrick, we'll get to the stoichiometry of this in a later slide. So this is the overview of what happens. You've seen this before. What does substrate level phosphorylation mean? It's the direct enzymatic transfer of a phosphate from a phosphorylated molecule to ADP to make ATP. And very often that phosphorylated molecule is called PEP, phosphoenol pyruvate. So it's pyruvate with a phosphate group attached. There's an enzyme called pyruvate kinase, which removes the phosphate group and gives it to ADP. So you end up with ATP and pyruvate. This term kinase, anytime you see an enzyme that has kinase in its name, it almost surely involves phosphate, usually transferring a phosphate from one molecule to another. And this shows you another view of how substrate level phosphorylation works. The enzyme here is that kinase. The substrate is a phosphorylated molecule. The phosphate is removed and transferred to ADP. So you get a product, pyruvate and ATP. And this is usually, as I said here, it's a one-to-one proposition. For every one molecule of PEP, you get one molecule of ATP. And now let's talk about oxidative phosphorylation. This is where we get most of our energy. It's energy from glucose by way of respiration in the mitochondrion. So electrons from organic compounds, usually glucose, are passed through a series of reactions called the electron transport chain to an inorganic molecule like oxygen. This oxygen is very important because without it, everything changes. Chemiosmosis generates the ATP as energy is released, and we get many ATPs from one molecule of glucose. I put this little diagram in, I've showed it over and over to remind you what chemiosmosis is. It's really quite a simple process where there's a high concentration of ions, usually protons, on one side of a membrane, low concentration on the other. Then the ions can move passively through a channel that contains an enzyme called ATP synthase, and that converts ADP to ATP. Chemiosmosis is defined as the movement of ions across a semi-permeable membrane down an electrochemical gradient. And in phosphorylation, the ion is usually a proton. So here's where we can count ATP molecules. And we'll start with glucose in the cytoplasm and glycolysis. So glycolysis results in, glucose is a 6-carbon molecule. It is hydrolyzed into 2 3-carbon molecules, which are called pyruvic acid. And these pyruvic acid molecules can make 2 acetyl-CoA molecules. They are the start of the Krebs cycle. So what's going on here? Well, as I said earlier, glycolysis itself yields 2 ATPs. Why is it 2? It's because you start out with 1 molecule of glucose, you get 2 molecules of pyruvate and an ATP associated with each, done by substrate level phosphorylation. Now, glycolysis also yields reducing power. Electron carriers, reduced electron carriers, 2 NADH plus 2 protons, conversion of pyruvate to acetyl-CoA, 2 more NADH, 2 more protons, and a carbon dioxide molecule is released. In this case, 2 of them at this step. Then in the Krebs cycle, 4 more molecules of carbon dioxide and a lot more reduced carriers, FADH2 and NADH, so that you're accumulating reduced carriers plus protons. These then feed into the electron transport chain, illustrated here. And I'll show you this more explicitly in a minute. But if you recall, I want to remind you what an electron transport chain is. And I refer you for an animated version of this. We looked at a video last time, and that video is in the recording. So if you want to go back and visualize how electron carriers work, take a look at that video. But in short, you have a reduced substance which transfers electrons to an oxidized one so that it is then reduced, and electrons then are transferred from that reduced carrier to another oxidized one to give another reduced carrier. And this goes on and on. So every one of these steps in the electron transport chain is another one of these toggles. And I show you that this again to make it more real to you what is the role of oxygen in all of this? Oxygen is the final electron acceptor. You see it here at the very end of the electron transport chain, 6 molecules of oxygen plus 12 protons yield 6 molecules of water. So the final electron acceptor, you have all these receivers and acceptors all down the chain. The last one is oxygen, molecular oxygen. That's why we breathe. That's why fish have gills. We have to extract oxygen from our environment. And if we didn't, if we couldn't do that, what would happen? So if there were no oxygen here, what would happen? Remember, electrons are passing down this electron transport chain. They get to the end. They have to go somewhere, but there's nowhere to go. So it stops. So under anaerobic conditions, if we run out of oxygen, the buck stops here. The electron transport chain stops. And if that stops, the Krebs cycle stops, and everything backs up all the way back to pyruvate. So I'm trying to emphasize the point that we are aerobic organisms, and the reason is we have to do something with our electrons in oxidative phosphorylation, and if we can't transfer them to oxygen, we're in trouble. Everything stops. So that's why I put in here the final electron acceptor is a half a molecule of oxygen giving a whole molecule of water.
Professor?
Yeah.
I forget where the question was, but there was a question recently asking if it was ions or electrons being transferred across. It's ions or electrons, correct?
Okay, we'll address that right here.
All right, thank you.
So we have, what's the beginning of electron transport from the Krebs cycle? If you go back here, Krebs cycle produces a lot of NADH and FADH2. So now we have the electron transport chain within the inner mitochondrial membrane. The source of electrons is NADH plus protons. So these enter the electron transport chain, which are these protein complexes shown here by globs embedded in the membrane. So the electrons are transferred from one carrier to the next, to the next, to the next, through the membrane, and losing energy as they go along. But look what else is happening. Protons are being produced. So the electrons are traveling along in the membrane. Protons are being concentrated outside the membrane on one side of it. So this is causing a proton gradient. And at some point, the protons are passed back through the membrane passively, this blue channel here. And when they do that, and they do it by virtue of the force of the gradient, the proton gradient, concentration is much higher on one side than on the other. ATP synthase converts ADP to ATP through chemiosmosis. This blue structure here is chemiosmosis. And that's where we get most of our ATP. Back here, most of our ATP comes from electron transport. So if you add them up, later I'll show you that it takes 2 ATPs to do glycolysis, but then it yields 2 ATPs. And so one way of adding them up is to say that this is 4. At the gross level it is, but the net of glycolysis is just 2. And then the Krebs cycle gives 2 more. Oxidative phosphorylation gives 34, which add up to 38. 34 here, and 2 here, and 2 here. So a 38 total of ATP molecules, and that is from 1 molecule of glucose. Where is this all happening? Here's a cell. Big blue thing is a cell with the organelles. And this little orange thing is a mitochondrion. You look inside it, you see that it is a membrane-bound organelle. It has an outer membrane. Inside, it has a membrane system called cristae. And it is composed of the inner mitochondrial membrane plus a matrix. So electron transport goes on within this inner mitochondrial membrane. And when electron transport occurs, it pumps protons into the matrix. And then when they're passed out into the intermembrane space, that's where chemiosmosis occurs and ATP is generated. So is this making sense? Following the process one step at a time?
Yes.
Okay. If you have any questions, let me know. So that spatially, this is where the action is. And now let's go back to the cytoplasm and take a more detailed look at glycolysis. Glycolysis is usually represented as though it's a one-step process. You have a molecule of glucose, has 6 carbons, and that is converted to 2 molecules of pyruvate, 3 carbons each. So it just cuts the molecule in 2. But what actually happens is a 9-step process. 2 of these steps, the first one, hexokinase, requires a molecule of ATP. There's another step farther down the path, requires another molecule of ATP. So it's actually using ATP to get started, but then it generates 4 ATP, 2 here, and then 2 just before pyruvate. So 4 ATPs are generated in glycolysis. It costs 2, so the net is 2. And this all occurs in the cytoplasm before pyruvate enters the mitochondrion. So this is called the grooming of pyruvic acid. A 3-carbon compound. So we go from glucose, a 6-carbon compound, to pyruvate, a 3-carbon compound, to a 2-carbon compound called acetyl-CoA plus carbon dioxide. So again, we're back in the cytoplasm. We have pyruvate from glycolysis. This is its structure. It's got these 2 red carbons plus a carboxyl here, which is the equivalent of carbon dioxide, CO2. And this carbon dioxide, this carboxyl, is liberated as the gas carbon dioxide. At the time pyruvate enters the mitochondria. And NADH plus H plus is generated. coenzyme A is added to this resulting compound, which is now a 2-carbon compound called acetyl-CoA. And this acetyl-CoA is the first step in the Krebs cycle. And the Krebs cycle is what generates much of the energy needed for ATP synthesis. So the next step after this is the Krebs cycle. And here you see it, acetyl-CoA, the 2 red carbons. The very first thing that happens in the Krebs cycle is that acetyl-CoA combines with oxaloacetate, which is a 4-carbon molecule, to make a 6-carbon structure called citrate, citric acid, found in high concentrations in citrus. So it makes citrus sour. Okay, so this is converted then to isocitrate, which just changes the configuration. But then a molecule of carbon dioxide is released. So we now go from 6 carbons to alpha ketoglutarate, which is 5 carbons. We still have our 2 red carbons here. And 1 blue one left. Then another carbon is lost through CO2, and more NADH is generated to get to succinyl-CoA, which is now a 4-carbon molecule. 4 carbons plus coenzyme A. The conversion of that to succinate generates ATP. So this is the substrate level phosphorylation that occurs in the Krebs cycle. And then this goes on, generates some more reduced carriers, and finally back to oxaloacetate, which is then ready to accept another acetyl-CoA and start the cycle all over again. So this cycle is going round and round, generating lots of reduced carriers and 1 ATP for every pyruvate or every acetyl-CoA that enters the cycle. So the thing to remember here is that the main function, the main product of the Krebs cycle is electrons for the electron transport chain. So here we have it. Again, we're talking about a cell, the nucleus, the mitochondrion, and oxidative phosphorylation goes on within the mitochondrion, but it starts outside the mitochondrion in the cytoplasm with glucose. And where did the glucose come from? Depends on depends the starting point. So if this is in a plant, where did the glucose come from? Think carbon dioxide fixation. That happens in the Kelvin cycle. So the Kelvin cycle takes the gas, carbon dioxide, and makes it into a solid, glucose. In an animal, this would not come from chloroplast, obviously, so it comes from the food you eat. So we get a substrate level phosphorylation, you get pyruvate, crosses the membrane, pyruvate, converted to acetyl-CoA, goes into the Krebs cycle, you get all these reduced carriers, which end up going into the inner membrane, the electron transport chain, and this is what the electrons are transported through the membrane, and as they go, they're pumping protons from the matrix into the intercellular space, intermembrane space. So a high concentration of protons builds up, making a proton gradient. And this then allows protons to passively diffuse through the membrane by chemiosmosis. Each of these little purple structures represents chemiosmosis, which is where ATP comes from. ADP plus inorganic phosphate makes ATP. Okay, so is that clear? The sequence of events that occurs here, and the sequence of important molecules: glucose, pyruvate, acetyl-CoA, reduced carriers, electron transport chain, proton gradient, ATP synthesis. Now, what happens here depends on the state of the organism. So if you have just eaten, say, a big meal, and your digestive system is making a lot of glucose, then this is a catabolic process, getting to acetyl-CoA. However, supposing you haven't eaten for 3 days, you're starving. So you have no glucose. What is your source of energy? It's fat. Fat, as you recall, is triglycerides. So if you have no source of glucose, triglycerides will start to be hydrolyzed to fatty acids, and fatty acids, by a process of beta oxidation, can be converted to acetyl-CoA. And then that starts the Krebs cycle, and you're back in business. You're making ATP, you're making reduced carriers, you're making a proton gradient, and you're making ATP. But now the source of the energy is coming from fat, not from sugar. But now supposing you eat a huge candy bar. So you have a lot of sugar, and you're overloading the system with sugar. So now you're making too much acetyl-CoA. Krebs cycle can't take it all. So the process to the right is reversible. It's actually anabolic, where acetyl-CoA can be a building block for fatty acids, and fatty acids can be converted into triglycerides, which is fat. So if you eat too much, you'll only use as much energy at the moment as you need, and any extra goes to fat. So this is a part of the process of the homeostasis within the cell, keeping the cell's metabolism balanced. If you're starving, you use fat to run the TCA cycle. If you've eaten too much, or if you've eaten the right amount, you run the TCA cycle from the food you eat. If you eat too much, you reverse the process and make fat. Everybody clear on that?
Yes.
Okay. So acetyl-CoA is called a crossroads compound, which I've just described. It's the high energy fuel molecule that starts the Krebs cycle. It's a common pathway point for extraction of energy from carbohydrates, fats, and proteins, in other words, from food. And in that sense, it's catabolic. It can also be used as a building block for synthesis of fatty acids, and in that sense, it's anabolic. So that's what I was trying to tell you in this slide. This pathway can run either forward or backward, depending on your diet. If your diet is just right, you end up just making ATP. If you're getting too much food, you're making fat. If you're not getting enough food, you're hydrolyzing fat. Catabolic pathways. The food we eat is largely composed of proteins, polysaccharides, and lipids, complex molecules. Most of the food we eat is not simple monomers. It's polymers. But these can all be broken down into monomers. So proteins can be broken down into amino acids, polysaccharides into monosaccharides, and lipids into fatty acids or glycerol. All of these can be converted to acetyl-CoA. And acetyl-CoA will start the Krebs cycle. The Krebs cycle then generates ATP, carbon dioxide, but most of all, most importantly, it generates reduced carrier molecules, which lead to the electron transport chain, chemiosmosis, and that generates ATP. So we end up with ATP from the complex food that we eat. Now also another polymer that we eat is nucleic acids, but it's a very small component of our food, so it doesn't figure prominently in the production of ATP. Starting with pizza. Pizza is a pretty complete food. It's got lots of fat, lots of carbohydrate, and lots of protein. The fats can be converted directly to acetyl-CoA, as can proteins through amino acids. Complex carbohydrates can be quickly converted to simple sugars, and then glycolysis, and we get pyruvate, which goes to acetyl-CoA, and that starts the Krebs cycle. So here's another look at the stoichiometry of aerobic respiration. It takes 2 ATPs for glycolysis to occur, but it generates 4. So you have a net of 2, and these are substrate level phosphorylations. There's another one of those in the Krebs cycle for 2 more ATPs, electron transport and chemiosmosis generates, in this case, 32 ATPs for a total of 36, or if you count these as gross, 38 ATPs. And this all occurs on the inner mitochondrial membrane. So now let's talk about what if you run out of oxygen, anaerobic respiration. It can occur in microbes like yeasts, or as we'll get to in a minute, it can occur in your muscles. So what happens? We have glucose glycolysis occurring in the cytoplasm. So glucose gets split into 2 molecules of pyruvate, and this normally would go on to the electron transport, the Krebs cycle, the electron transport chain, but there's no oxygen, there's no Krebs cycle. So what happens to the electrons? Pyruvate loses 2 molecules of carbon dioxide. So you lose this carboxyl group so that you go from a 3 carbon molecule to 2 carbons, which are called acetaldehyde. And acetaldehyde then is hydrogenated to ethanol. 2 protons from glycolysis are added on to acetaldehyde here and here to make 2 molecules of ethanol, for example, wine. So when there's no oxygen, there's no Krebs cycle, and pyruvate goes, instead of the Krebs cycle, it goes to acetaldehyde and then to ethanol, and that's the end. Now, what if it happens in our muscles? Same thing, glucose in the cytoplasm, glycolysis, 2 ATPs are generated, but it stays in the cytoplasm. And now the pyruvate that results from glycolysis is hydrogenated directly to 2 molecules of lactic acid. So there's no carbon dioxide produced. You end up not with a 2-carbon molecule, but 3-carbon acid called lactic acid. So no oxygen, no Krebs cycle, no carbon dioxide, just a straight conversion of pyruvate to lactate. So this brings us to the issue of vigorous exercise, which leads to anaerobiosis. You have glucose in the cytoplasm of muscle cells. It's converted to pyruvate, which generates 2 ATPs and lactate, which then can get cycled through the liver back to pyruvate and glucose. So this cycle can go on. But what happens if you are engaging in high intensity exercise? Your muscles run out of oxygen because they're demanding oxygen faster than your lungs can supply it. So your muscles become anaerobic. But they need ATP because they're working hard, they need energy. So they can do one of 2 things, either get ATP from anaerobic glycolysis, which I just showed you, but very limited amount. They get 2 ATPs for every molecule of glucose that's being hydrolyzed, only 2 ATPs, but at least it's something. The alternative would be to wait for more oxygen, wait for your lungs to deliver more oxygen to muscle cells. But depending on the demand, if the demand is high, they can't wait, and so they generate ATP and lactic acid, and that's what causes your muscles to burn. It's the accumulation of lactic acid as a result of running out of oxygen in your muscles. So is that clear why? We've all experienced this. Anytime you push yourself hard enough, your muscles will burn. And now you know why. This shows a side-by-side comparison of what happens in yeast, what happens in muscles. In yeast, it's a 2-step process from glycolysis, from glucose to pyruvate to acetaldehyde to ethanol with the release of carbon dioxide. You go from a 3-carbon to a 2-carbon to a 2-carbon. Whereas in muscle, you don't have this intermediate step acetaldehyde. You go directly glucose, pyruvate, lactate, with the release of the ATP that your muscles so badly need. So the overall chemistry of respiration, a glucose molecule plus 6 molecules of oxygen, O2, will yield 6 molecules of carbon dioxide, CO2, plus 6 molecules of water, plus 38 molecules of ATP. That's the overall chemical reaction. And this is simply a review of what we've been talking about. 2 substrate level phosphorylations, one in glycolysis, one in the Krebs cycle, and then a big production of ATP as a result of electron transport. And where do the electrons come from? Mostly from the Krebs cycle. Krebs cycle yields electrons for the electron transport chain. That generates a proton gradient, which allows chemiosmosis to produce a lot of ATP. So let's go over the steps of glycolysis again. It's exergonic series of reactions. It's catabolic. It occurs in the cytoplasm outside the mitochondria. The glucose oxidation process begins by breaking glucose into 2 molecules of pyruvic acid. So remember, glucose is a 6-carbon sugar. It's broken up into 2 molecules of 3 carbons each, pyruvic acid. And that makes 2 molecules of ATP from every 1 molecule of glucose. So that's why it's so inefficient in your muscles in anaerobiosis, is that you're relying on glycolysis only, not oxidative phosphorylation for your ATP. And glycolysis is a low yielder of ATP. The overall chemistry of glycolysis, you have a 6-carbon glucose molecule. It's split into 2 3-carbon pyruvates, yield of 2 molecules of ATP and 2 NADH plus 2 protons. We've gone through this before, don't need to do it again. It's simply the 6 steps of glycolysis, 9 steps of glycolysis. And the first, at the beginning of the pathway, it actually uses ATP. It uses one in the first step with hexokinase, and another one, the conversion of fructose 6-phosphate. But then at the end, it generates ATP. 2 ADP makes 2 ATP here. And over here again, at the next to the last step, just before pyruvate, you get another 2 ATPs. So the stoichiometry of it, glycolysis produces 4 ATPs. It costs 2. So the net is 2. And they all occur at the level of substrate phosphorylation. And finally, electron transport chain and chemiosmosis. This summarizes the entire process from the cytoplasm through the mitochondrial membrane to chemiosmosis to ATP. And you can see the critical step here is the generation of this proton gradient, and importantly, having oxygen available to be the final acceptor of the electrons that come down the electron transport chain. And the source of the electrons, of course, is the Krebs cycle. Most of them come from the Krebs cycle. Okay, that's the end of the slides, and we're just about out of time. Anything that you want to talk about before we go?
I have a question, Professor.
Yeah.
About the group projects.
Yeah.
So my group, we've been in contact with each other and we've been working on stuff. My group is starting to feel like one member may not be contributing as much as they should. If by the time we do our rough draft presentation to you, which we already have scheduled. If by that time we feel that this person is not going to be contributing efficiently or sufficiently enough, can we boot them or?
Yes.
Okay.
But before you do, let me send a message indicating that time's up. That
Okay, yeah, it's just like we have a group document that we've been working on, and two of us have put in quite a bit of stuff, and the other person has put in not much and doesn't really seem to be understanding the concept. So we're gonna get together tomorrow and see if we can remedy that. But if it's not fixable, I guess we'll let you know.
Okay, I'll hold off then until after. You're going to have a meeting of the three of you, right?
Correct.
Okay, I'll wait till after that meeting. Let me know the result, and if necessary, I will send a message to all three of you indicating what's going to happen next.
Thank you.
Okay, anything else? All right, remember there's a quiz on Wednesday. You have all day to take it from midnight to midnight. And it's on photosynthesis and respiration. Okay, if nothing else, I'm going to stop sharing. And I will see you all on Wednesday. Bye.
Thank you, Professor.
Mhm.
Thank you.
All right, bye.